Assalamualaikum and a very good day. My name is Imein Sufia Binti Mohd Adly from Group RAS 2016A. Today, I will be presenting my scientific research report entitled The Identification of Escherichia coli from Chicken Samples Using Culture-Based Method and Bioinformative Tools for HLYF Gene. This presentation will include introduction, literature review, methodology, results and discussion, and conclusion and recommendation. Zoonotic avian Escherichia coli, known as APEC, is a major pathogen within the poultry industry. This is because APEC has the ability to contaminate chicken products and infect humans. However, colibacillosis, which is a disease from chickens, is caused by a variety of E. coli strains with pathogenic characteristics similar to APEC. From this research, the virulent gene HLYF was chosen as the gene of interest of APEC. The problem that has arisen in this research is living chickens can harbor pathogenic bacteria and deteriorate the human health. Poor handling can result in contamination towards consumers. Next, bacteria is commonly identified via biochemical testing, which is time-consuming and less sensitive. Therefore, this study uses bioinformative tools such as BLAST-N, CLUSTAL-W, and in silico PCR for the molecular detection of the HLYF gene. The objectives of this research includes 1. To isolate APEC from unhealthy living chickens, and 2. To design specific primer pairs of virulent gene HLYF from the genomic sequence of APEC. E. coli is a gram-negative, rod-shaped, facultative bacteria belonging in the Enterobacteriaceae family. APEC is both a primary and secondary pathogen of chickens and other avian species. Broiler chickens and turkeys are some species that are susceptible to APEC infection. APEC causes extra-intestinal infections, which are the main causes of respiratory infection, pericarditis, and septicemia in poultry. APEC's mode of transmission is invading the gastrointestinal and respiratory tracts and reaching the bloodstream and internal organs. Chickens can also be infected from contaminated food and water, fecal-oral, and aerosol routes. 30 bacteria samples obtained from unhealthy living chickens were collected via cloacal swabbing. The swab was gently inserted into the chicken cloaca and rotated about 5 times clockwise and anti-clockwise, and placed in the centrifuge tube containing the viral transport medium. The samples were kept in an ice bucket until further use. For the preparation of nutrient broth, 4 grams of powder was suspended into 500 milliliters of distilled water. This step was repeated again to obtain a volume of 1 liter. The mixture was placed in 2 500 milliliters of Scott bottles and placed onto a hot plate stirrer until fully dissolved. The mixture was autoclaved at 121 degrees Celsius for 15 minutes and stored in the fridge at 4 degrees Celsius. McConkey agar was prepared by weighing 25 grams of powder and adding 500 milliliters of distilled water. This step was repeated again to obtain a volume of 1 liter. The mixture was placed into 500 milliliters of Scott bottles and placed onto a hot plate stirrer until fully dissolved. The mixture was autoclaved at 121 degrees Celsius for 15 minutes. The agar mixture was poured into agar plates in the laminar flow chamber and sealed with parafilm. The plates were stacked upside down in plastic bags and chilled at 4 degrees Celsius. The preparation of nutrient agar is similar to McConkey agar, where it only differed in weighing 10 grams of powder and 500 ml of distilled water. E. coli subcultures were prepared by thawing the nutrient broth for 1 hour in the incubator. 9 milliliters of nutrient broth was placed in a labeled biju bottle. This step was repeated another 29 times. Next, 1 milliliters of the sample was poured into the bottle, gently swirled, and incubated at 37 degrees Celsius for 18 hours. Next, the subcultures were streaked on the McConkey agars using streak-plick method. A septic technique was applied to prevent contamination. Streaking on nutrient agar was also similar to McConkey agar. After streaking, the plates were sealed with parafilm and incubated at 37 degrees Celsius for 18 hours. After 18 hours, gram staining was done. The bacteria colonies on the plates were inoculated and smeared onto a clean slide and heat fixed with the Bunsen burner. Crystal violet was added for 30 seconds and rinsed with distilled water. Iodine was later added for 1 minute before rinsing with distilled water. Wash off with 95% of ethanol for 10 seconds, followed by distilled water, and finally, safranin was added for 10 seconds before rinsing with distilled water. The slides were blotted and observed under a light microscope. Malonate broth was prepared by suspending 4 grams of powder into 5 milliliters of distilled water. The next steps were similar to the preparation of nutrient broth. Malonate test was performed by measuring 3 milliliters of malonate broth into a centrifuge tube. The 19 positive McConkey plates were inoculated using a glass rod and stirred into the tubes. The tubes were loosely capped and incubated for 24 hours. 20% of glycerol stock was prepared by adding 200 milliliters of glycerin to 800 milliliters of distilled water. Once autoclaved, 5 milliliters of subculture and 5 milliliters of stock were added into 19 centrifuge tubes and slowly shaped. The tubes were labeled and kept at minus 20 degrees Celsius in Ziploc bags. DNA extraction was performed by following the steps shown in the slide. For primer design, the full nucleotide sequence of HLYF gene was obtained from gene bank and compared with other similar sequences using BLAST-N. The similar sequences were later aligned using Cluster W in BioEdit software. Forward and reverse primers were designed based on the unique region of the HLYF gene using Primer 3 program. In silico PCR was used to test the specificity of the gene. Agarose gel electrophoresis was performed as shown in the slide. Out of 30 samples, 19 samples were McConkey positive, where pink colonies were formed. The remaining 11 samples were negative and formed opaque off-white colonies. Some plates exhibit a mixture of pink and white colonies. This happens when the media becomes alkali and unable to ferment lactose. From the gram staining, all 19 positive plates appeared pink, indicating the presence of gram-negative bacteria, which is E. coli. There were also some purple stains present because bacteria isolation was not yet performed on the fresh samples. Malonate test was performed to test the ability of E. coli to use malonate as its main source for carbon. For E. coli detection, the malonate broth should remain unchanged, which was a green color broth. The results from the malonate test showed all 19 positive samples were malonate negative, maintaining the green color. Once the biochemical tests were performed, the 19 confirmed positive samples were stripped onto the nutrient agars for pure culture and to obtain single colonies. The pure cultures were kept in 20% glycerol in microcentrifuge tubes for archiving purposes and stored in minus 20 degrees Celsius to stop growth. DNA extraction was done to purify the DNA samples using the Real Genomics DNA extraction kit. All 19 samples produced a pellet. Primer design consisted of 5 steps. First was generating nucleotide alignments using BLAST-N. The sequence obtained from accession number AY545598.5 was 100% specifically targeting HLYF gene, which indicated it as a highly conserved gene. We do not choose identity less than 100% because in some organisms, 99% identity may suggest that it is another species or genus and might not be conserved. The nucleotide sequence was later saved in FASTA format. FASTA alignment is responsible to search for the similarities between 2 or more DNA sequences coded for HLYF gene. The size of gene obtained was 1,753 base pairs. Cluster W is a program used to perform multiple sequence alignment to find unique regions between other species. The HLYF sequence obtained from E. coli plasmid P. APAC02 called V was aligned with another E. coli strain BEN5202 and Salmonella enterica subspecies. The forward primer was between nucleotide 1473 to 1492, while reverse primer was between 1681 to 1700 at 222 base pair. Both primers were considered strong because both contained 20 bases with 40% and 45% of GC content respectively. The following equation shows the calculation of GC content. In silico PCR is a virtual technique developed for nucleic acid amplification technologies. In silico is done using a computer software to predict the outcome of the real PCR reaction. In silico is also used to test the design primers. It can be seen that the design primer pairs successfully coded for Escherichia coli APAC01 and produced a bending pattern with the amplicon size being 228 base pairs. Lastly, gel electrophoresis was performed to separate and analyze the DNA and their fragments based on size and charge. Gel electrophoresis is usually done for analytical purposes, often after the amplification of DNA via PCR. DNA travels through the agarose using an electric field. The molecules travel across the gel at different paces, thus forming distinct bending patterns on the gel. The first and last lane was placed with 1 kilobase pair DNA ladder and Hin3 DNA marker respectively in the 1% agarose gel wells. The positive controls, lane 3 and lane 5, showed distinct bending pattern at 228 base pairs, confirming with the result obtained from in silico PCR. Lane 2 and Lane 4 produced no bending patterns because they were the negative controls. In a nutshell, this research successfully achieved the 2 objectives earlier. APAC was successfully isolated from the cloacal swabs of 30 unhealthy living chickens via biochemical testings such as streaking on the McConkey agars and malonate test. Next, the specific primers of HLYF gene was successfully designed artificially using bioinformative tools such as BLAST-N, FASTA, Cluster W, and in silico PCR. Agarose gel electrophoresis showed the bending pattern of HLYF gene. which is complementary with the in silico PCR result. It is recommended that PCR to best be performed in the lab. This is because PCR is not time-consuming and still manage to provide a rapid, specific, and sensitive result. The result obtained may be more accurate rather than using artificial intelligence, thus providing a more clear bending pattern when gel electrophoresis is performed. This way, not only does it benefit the consumers, poultry industry, and other food industries, it can also prevent further disease contamination in food preparation and avoid country recession.